Yeah, so I was asked to talk about what I think are the most important next questions in aggressive B-cell lymphoma. And I focused on four main things. I think we're seeing a lot of new things in large cell lymphoma. We're seeing new ways of curing patients. And the idea is, how to actually increase that cure rate even further. It's clear we're in the era of immunotherapy. We now know that CAR-19 therapy cures patients with large cell lymphoma. And what we're going to learn relatively soon in randomized trials is, does the cure rate go up if you actually strategize using CAR-19 as part of frontline management? So that's a very important study. The first one is called ZUMA-23, and it's going to challenge anthracycline-based chemotherapy. So this is a very important question, and it's not at all clear what's going to be the winner. Anthracycline-based chemotherapy still cures a lot of patients, and so we need to know if that's going to improve the cure rate. But other immunotherapy agents are also showing that they can probably cure patients with large cell lymphoma in the relapse setting. At least the duration of complete responses seem to be long enough to suggest that. So they are being also tested in the upfront setting. The biggest difference is they're being added to chemotherapy. So the idea is, can you add these drugs to standard anthracycline-based chemotherapy and cure a substantially increased number of patients? So that's really question number one. I will highlight that that's in patients across the large cell lymphoma spectrum. Another important question is, can we select subtypes of large cell lymphoma that benefit from certain agents? And that's being tested in a number of studies, one of which is the Escalade trial, which is testing the addition of acalabrutinib to R-CHOP compared to R-CHOP plus placebo. The biggest difference is, at the beginning, the patients are selected for their status, whether their cell of origin status, and if they're a non-GCB tumor, they're eligible. So this builds upon decades of biology suggesting that these genetic subtypes do better with different agents. So we'll actually learn that. The third question, which is really exciting, is maybe we can even cure these patients without chemotherapy at all. So our group has recently published a paper that suggested that combinations of targeted therapies actually probably cures some patients that have relapsed after chemotherapy, and even some of them after CAR-T. So the idea is, if that's true, is there a way to use these targeted agents in a smart way without any chemotherapy and possibly cure patients up front or in the relapse setting? So those are exciting therapy questions. The last thing I highlight in the talk is that we're also learning on the best way to define remission. So what's zooming ahead is the use of these assays for measurable residual disease, mostly circulating tumor DNA. These are tools we can use to help us understand who's had the best response to therapy. But in aggressive lymphomas, it might actually show us who needs additional therapy because they aren't actually cleared yet of their MRD. So those studies are also ongoing. So there's a lot ongoing in large cell lymphoma.